All right. Hello, everyone. I am your last talk between you and the break. But I think Laurel did an amazing job of introducing anatoxins in rivers. And that's something that our lab, so Dre is here. She's going to be working in the Scott River watershed, building off of Laurel's work. And another way that we're building off of actually some of Laurel's previous work is we're thinking a lot about how can we actually predict the timing and the magnitude of these anatoxins in rivers? So Jordan is leading this work, but she wasn't actually able to join us after all today. So I'm going to be giving a version of her talk. So I just want to start briefly with acknowledgements. We've had a lot of awesome support, both from our previous project collaborators, the Karuk Tribe Mentoring Committee that we established, as well as other members of the lab. All right, so I also have a little bit of a text conversion thing going on, so it's gonna be weird, but it's fine. All right, so given that Laurel sent me her slides ahead of time, and I was like, awesome, you're giving them the full picture of sort of the ideas that we're thinking about when we're thinking about HABs and rivers, and specifically benthic HABs and rivers. And so here's Laurel about 6 years ago looking at some macrophytes in the Klamath main stem, and she took me out and got me really excited, as you saw, just a couple of minutes ago, feeling really inspired about the certain level of discovery that's left to do with these benthic cyanobacteria and how it's actually a really amazing intersection of public health issues as well as ecological health, right? And so studying these, I felt really inspired. And when I got back to my office, I was just starting my job at the University of Nevada, Reno. And, Laurel was saying that at a global scale, these benthic HABs are pretty sparse. But in my mind, actually 10 countries having tons of anatoxins in their rivers, to me, that sounds like a lot. And it sounds like a big problem because it probably means that they're in other places that we just aren't monitoring. And so as Laurel pointed out, right, these are also increasingly being found. Oftentimes, people don't even know that there's benthic HABs in rivers until a dog unfortunately dies, and then that spurs a lot of research. Although we know that anatoxin concentrations tend to peak in the late summer and fall, whether they peak in July or whether they peak in October, that can make a big difference for whether people want to recreate in that river, right? And so having that understanding for the public and being able to predict that could be a really useful tool for managers. And then it could also be a useful tool for ecologists that are thinking about, let's say, the salmon and wanting to understand, are these going to have a certain effect in a river? So at the same time that Laurel and I were spending time on the Klamath River, I was also sort of starting to learn a lot about building predictive models. And so this is going to be, I don't want to overpromise in this talk. I'm more giving you a vision of where a lot of the research in our lab is going to be going over the next 10 years or so. But our ideas are to use predictive models, similar to the talk that we saw with Claire, right? Thinking about flow. So this is more thinking about population dynamics in rivers. So here, gross primary productivity, this is a metric. You can use high frequency sensors and look at changes in dissolved oxygen throughout the day, and you can estimate the amount of sort of community productivity that there is. So how much algae was growing in the river at that time, how much sort of breakdown of that algae was occurring, right? And by using population models and sort of fitting those models to data, we can also start to estimate the magnitude of the underlying biomass. So the idea here is, okay, are we going to be able to maybe take some of our ecological understanding, use our sort of mathematical models that we have for population dynamics, and then be able to predict anatoxin? And again, the sort of overarching motivation for this is that there's been a lot of sort of discussion in the ecological community about how weather forecasting. We all love the fact that we know what the weather's going to be like in 4 days, right? And so can we use those ideas of incorporating sort of weather forecasts, incorporating forecasts of flow and temperature, and then use our ecological understanding to then be able to do near-term forecasting. So this is on the order of months to weeks. So in this work that Jordan's leading, we're really in these beginning stages. We're sort of understanding how this might look like, what is the structure of the models, and we're obtaining that data. Whoops. So that's all right. Our end users, something I forgot to say, is really, I really like this sort of conceptual figure of what this forecast might look like, because it walks you through the stages of sort of understanding how this might look like. And so one of the first steps is, of course, engaging end users and thinking about who that might be. And so this came up a little bit in the question and answer portion, right? But of Laurel's talk, but it's thinking about, okay, who are, maybe it's drinking water managers, right? Who are thinking about, oh, are there going to be anatoxins in my river? Maybe this is people who are posting signage, right, around rivers and helping the public understand the risk. Yikes. But of course, the challenge is how are we going to actually predict these anatoxins? Because the thing is that there's been quite a bit of work out of New Zealand, right, that has shown that there's not just one main predictor. And when we look across many different river sites, so this is for Midium, you can kind of think of this as synonymous with microcoleus. It used to be classified that way. You can see that different rivers have different peak timing of anatoxins, right? And so there's a lot of spatial variability. And then in addition to that, among years, we were bringing up, if it's a good water year, maybe we're going to have lower anatoxins that year versus higher anatoxins. This is some of Keith Boomer-Gregson's work out of, oh, sorry, yes. Oh, for the people in the back. Okay, sorry. All right, I'll use my teaching voice. Okay, yeah, so as I said before, right, there's not just one silver bullet predictor. These anatoxins are affected by a lot of different things. So here is a figure from a New Zealand study where they basically showed that the highest biomass of these anatoxin-producing benthic cyanobacteria are in faster flowing rivers. They also tend to occur with higher temperatures. Nutrients have a role. It's actually quite complicated, but that's another predictor that we might be able to use. And then Laurel brought this up, but there's also these sort of complicated relationships where in some cases, there's more anatoxin production with more percent cover, but also there's contradictory studies, right, that show that there's no relationship in their studies, right? So there's still a lot of relationships we have to resolve. But most of those predictors that I just listed were abiotic predictors. So they're physical or chemical drivers. But something that hasn't really been thought about before is how can we maybe incorporate some of our biology knowledge in that GPP? If we understand some metric of the entire community dynamics, can that help improve our predictions of what's going on with the benthic cyanobacteria? And what's great about this when you're thinking about specifically a forecasting situation is that the USGS and then also lots of other people, right, have dissolved oxygen sensors that are distributed in rivers around this region, but also around the globe around the country. And so if you're able to then use that biotic understanding of the whole ecosystem using high frequency sensors, it's possible you could use that information to then forecast, again, the toxins. But before we do that, right, we have to understand what are those relationships? What do they look like? And so that's really what Jordan's looking at right now. And we kind of have these three hypotheses that address the question of to what degree do processes that shape overall productivity dynamics, which is also not just predicted by one thing, but many, how do those predict the occurrence and dynamics of toxins producing cyanobacteria? So you can imagine if they're all under the same predictors. So here we have just a conceptual figure where blue is flow. So we're thinking about a Mediterranean climate here. Green is a hypothetical sort of curve of the amount of productivity in a river over the summer. Purple is this anatoxin concentration, and yellow is the cyanobacterial occurrence. So they may be synchronous. It's possible that cyanobacteria lags that overall productivity in the river and that the anatoxins follow the relationship of total percent cover. Or there's been some work in the lab that has basically shown that that under certain conditions, it's possible that anatoxin concentrations peak in those early stages of mat development. So these are kind of our alternative hypotheses that we're looking at. So although this is the Klamath Basin Monitoring Program meeting, I'm going to talk about a broader range of rivers that we surveyed, in part because we ran into some issues with the sampling and fires, as I'm sure you guys are familiar with. So anyways, in 2022, Jordan sampled biweekly at the Salmon River, the South Fork Eel, and the Russian River. And then because of what we found in the Russian River, which is mostly that the anatoxins weren't peaking in our time frame, we switched over to weekly sampling of the South Fork eel and still doing biweekly of the salmon river. So the idea is that we have these dissolved oxygen sensors, and then in the oxygen footprint upstream that it's capturing, we monitored, took samples of the benthic mats, and we took water samples, took a bunch of other measurements. We brought that back to the lab. Taryn and Jordan did a ton of microscopy to identify those samples and look at the community composition. We also sent off samples for LC-MS analysis. So basically you can get really fast toxin concentration information from Tim Otten, who's great, right? And we'll send you back that sort of total anatoxin information. But anatoxins actually have a bunch of different sort of forms, different congeners, and those different congeners have different levels of toxicity. So for the purposes of our study, we're trying to look at the shifts in those the composition of the toxins as well. And then we did the metabolism modeling that gets us that whole community look. So this is kind of what it looks like, us going around, picking up rocks, looking at some cool microcoleus. So I'm just gonna show you some very preliminary results. Emphasis is on preliminary because there's been sort of a lot of challenges there, but I'm gonna first show you our GPP data. So again, this is sort of an indication of whole ecosystem community productivity. So we have the South Fork eel on the left-hand side, then the Salmon River, and then the Russian River, which again, we only sampled in 2022. So these are daily estimates of gross primary productivity. We had sort of a storm event that knocked back the community back, and it sort of recovered back in 2022. But you can see on the whole, if you just look across these, don't look at any individual day. Jordan's still working on sort of cleaning up the data for things that we feel uncertain about. But what you can see is the GPP dynamics are unique to each river in each year. And so similar to that New Zealand study, right? You know, just saying that, oh, anatoxins generally peak in the late summer and fall doesn't actually really capture what's going on in each of these unique systems across years. So now if we look at percent cover, so here, it's kind of hard to tell them apart on the screen, but this is green and this is sort of orange, yellow. We're looking at percent cover across years. And one thing I want to point out, unfortunately for the Klamath Basin, this part of our study in the Salmon River, we had issues with accessing our sites for fire reasons. And so I'm just going to show you all of the sites. But, and if you specifically focus on the South Fork Eel River, what you'll see is Anabaena, which is in green here, and microcoleus. Anabaena tends to peak a lot earlier than microcoleus. So Anabaena is another cyanobacteria. Molecularly, apparently it can't produce anatoxins itself, but it has other cyanobacteria living in it, whether that's microcoleus or Gietlerinema, I can never say that one, that is producing anatoxins. So we typically find relatively high concentrations found in those mats. But anyways, what this shows also is that this is showing you that just because microcoleus is maybe peaking at a certain time of the year, that doesn't mean that your anatoxin sort of risk for the public is just occurring then. It can extend over a longer time period in the season. So it's important to think about the full profile of cyanobacteria in your river. All right, and so finally, I'm gonna end on a lot of question marks. This is very unsatisfying, I agree. But with LC-MS analysis, we've had a very generous collaborator who's been running our samples, but he's an emeritus professor. He doesn't have a lot of technical support, and so it's taken a while. So we don't really have that data. We have some preliminary data that we got with help from the California Water Board that would maybe indicate that we have this lag in anatoxin concentrations, but I don't want to say anything conclusively there. So in summary, though, I want to talk about two sort of logistical points about sort of our work and thinking about if you were to create a forecast for your river. One thing is that, of course, collecting samples over time in fire-prone regions is challenging, right? And so, you know, our expectations of how well we can characterize systems through time needs to take in those sorts of considerations. But, another thing is that, you know, depending on your goals, you may think about how you analyze your samples differently. So for us, we really want to have that LCMS-MS data to look at those different congeners. But if you're thinking about this from a management perspective, obviously I would talk to Laurel. She's an amazing expert at this, but I would probably go either the route of sending it off for ELISA analysis, or as Laurel said, if qPCR can sort of replace our need to do ELISA, that is also an awesome option and potentially a cheaper option. And finally, we have other data that I didn't quite show, but basically we have some preliminary evidence. And once we finally have our toxin concentrations, we're going to be able to sort of explore these relationships and use population models to try and see how well we can predict our dynamics. But there's some evidence to suggest that there's this lag in the timing of the toxin concentrations. And we are continuing on our time series at the South Fork eel to try and get at that interannual variability question. So that's where we're at. And yeah, and then this is a note from Jordan. Jordan and Taryn are both talented artists. They spend a lot of time looking under a microscope. And so they're going to be creating a little coloring book for the Salmon River algae. And we're going to share that with anyone who's interested. Thank you.
All right, thanks, Joanna. Questions from the room?
Yeah, so this is more of a comment than a question. I have 8 sites across the Scott that we have been monitoring for the last 3 years for DO, temperature, biweekly nutrient sampling. We did a year of CRAM assessment on all the sites, so I have data to offer to support whatever work's going to go on in the Scott.
This is amazing.
Most of them are on public land too. So
that's awesome, especially, so Dre is going to be, she's going to be thinking more spatially than Jordan has. And so we're trying to actually hone in on which sites we're going to be monitoring, and preliminary data would be really awesome for that. So thank you.
Yeah, the data set's pretty good. And we're going to continue monitoring them in the future because the Scott's impaired for biostimulatory conditions, which is basically primary productivity issues. So
yeah, that's great. Thank you.
Other questions? All right. Laurel?
Did you guys look at, like, with your rock scrapes, like what proportion of the algae that you're just encountering in your sort of like transect surveys are actually cyanos and or microcoleus compared to like other taxa that are not toxin producers?
Yeah, it's super small for the most part, right? And so we, I don't know if Jordan talked to you about this, but we did sort of a following the swamp protocol, right? Did more of a random sampling of the full community. And then we did more targeted collection of the mats in addition to that. And I would say the relative proportion of cyanotoxins or cyanobacteria is relatively small. So it's definitely not New Zealand, but that's where trying to incorporate that into the models and maybe have it be more of like a prediction of, not necessarily, this is something we're going to be testing, right? Is like, I don't know how well we're going to be able to predict the like extent of the mats, more of like presence, absence. Is there a higher likelihood of it occurring on a particular date? But that's, yeah, definitely something.
And are you seeing them associated with like other taxa, like other types of algae, like, or do you have the data that could be?
Dissertation.
Okay.
Yes,
Nice.
yes.
Awesome.
And one of the things that we're looking at that I think is going to be something really cool, and I have so many ideas for further pursuing this, but you know, like you discussed, right? So anatoxins are these nitrogen-rich compounds, but the microcoleus grows in a lot of nitrogen-limited systems. And so where are they getting the nitrogen needed for the anatoxins? It may be that they're co-occurring in the same mats with nitrogen fixers, but who knows, right? How leaky is that nitrogen? You would have to do some isotope tests, I think, to look at like the mass transfer into the microcoleus and then actually look at, do we see the sort of nitrogen from the nitrogen fixers in the anatoxins itself? Yeah.
This is more of a comment than a question. I'm from the Salmon River, and just thinking about your issues regarding wildfire access, was that related to forest closures for public safety, or was it just kind of smoke and your choice for health? And
some of it was road closures.
Okay.
And some of it was smoke for health for my students. Yeah.
So when it comes to closures, I just wanted to make a comment. You know, working on the Salmon River, we have wildfire closures almost every summer, and we wouldn't get our work done if we had to always respect closures. I mean, we do respect closures, but there is a process to apply for a like a waiver for access for a closure, and it's
great.
relatively simple.
Yeah.
We do it for our staff and contractors pretty much every summer.
Great.
And as an ongoing issue in our basin, but also other places in California, when it comes to research and monitoring, it's worth kind of tuning into that process and you know, being aware when fires are, you know, happening, that closures tend to default to being very broad, and large areas of the closures, you know, you know, you can access them without actual risk to for your safety. So,
no, I think, I mean, that's a great point. And, you know, I am new to this area just in the last couple of years. And so I think in this initial phase, these are just the challenges that we face. But I agree, it would be, we're not going to be continuing sampling on the salmon. The funding for this is ending, but I think in the future we will be.
It's an issue on every river in California.
Totally,
so,
everywhere. Nevada.
Coloring book is, we would love to get some for our watershed education.
Yes, that would be fantastic. And
thank you.
So getting to the point in the project where we're thinking about what are the materials that we can create that are going to support sort of educational activities and outreach. And Laurel and I have actually been working on that a little bit together as well. So we would love to connect. That'd be awesome. Thanks.
All right. Any other questions in the room? All right. Well, Joanna, thank you so much. And thanks to all our presenters.